Hello, folks. So, the topic today is learning, and here we are interested in terms of real world phenomenon of when do you succeed and fail in changing your behavior. So, let's say it's New Year's Eve and you've made some resolutions. I'll do this, I'll do that, I'll exercise more. But a couple of weeks into it, you sort of start slipping back into your old habits. Why is it that when we have a lot of reasons that we're thinking to change our behavior, we nonetheless sometimes fail to change it? It's these kinds of aspects of behavior that we are primarily interested in today in terms of learning. Research on specifically behavior change and learning is strongly influenced by a psychological sort of era and set of principles which were called behaviorism. And behaviorism was a sort of reaction to the early introspectionist approach, um, in which there was a attempt to understand everything about psychology, including behavior, by looking at subjectively felt um, conscious experiences. And as you remember, that did not lead to a great deal of success. So, um, psychologists shifted to trying to understand behavior by and looking at observable events like what stimuli or situation the behavior was occurring in, um, what behavior was shown, and dropping out an attempt at theorizing what's going on in the middle in terms of consciously felt experience. And this sort of technique and emphasis was what we call behaviorism. As you may remember from the first module, um, behaviorism really zoomed in on um conditioning, which is to say two kinds of associative learning that seem to be operating and describing and predicting links that occur between environmental stimuli and the patterns of responses or behavior that people show in those situations or in terms of those stimuli. These days, of course, um, we would also, in the cognitive approach, include um, cognition, thoughts, expectations, and knowledge, but we'll return to impacts of these knowledge states and thoughts a little bit later. We'll first look at the pure associative learning techniques and pure associative learning mechanisms that were identified and described, and then extend our understanding a little bit further. So the first of these is classical conditioning. And this is associated with the work of Ivan Pavlov, um, who's a Russian psychologist. Later on, of course, he lived in the Soviet Union. He was primarily a physiologist. He was interested in the machinery of the body. In fact, he got his Nobel Prize on the digestive system. Um, and his work in associative learning or classical conditioning and arose from his study of digestion. What he was doing was he was um, looking at the role saliva plays in digestion. So he was using dogs and he had made incisions in the salivary ducts of dogs and he inserted a tube. And dogs have a reflexive or instinctive response to food, which is when they smell and taste food, they start producing saliva. So the dog would be given food, the dog would start producing saliva. Pavlov would collect the saliva and chemically analyze it. What enzymes does it have? What role does it play? Then he noticed something curious happening. He um, saw that the dog started producing saliva at the site of Pavlov or at the site of the dish in which the dog was given food. Initially, Pavlov was just thought this was a problem to be solved in terms of get rid of it. But then he realized that he was looking at the phenomenon of associative learning. If you think back again to the first module, where we talked about the British associationists, and they had talked about the mind being a blank slate and only having in the beginning an ability to link stimuli that were occurring together. And the mind links together stimuli, and that forms an idea. And as an organism keeps on experiencing the environment, more and more ideas get built up. And Pavlov thought, I've got the machinery of association at work here, and this is a very powerful way to study it. The initial speculation in terms of philosophy had been about subjective joining of sensations, ideas inside the head. But Pavlov saying, well, when the dog can't describe, can't introspect, but the dog is producing saliva. Um, Initially, it had no saliva to seeing Pavlov. It's not a built-in reflex. This is a learned reflex, or as Pavlov called it, it's formed a conditional reflex. So his term conditional was translated as conditioning, and that's what this has come to be called conditioning or Pavlovian conditioning or classical conditioning. So he built a separate lab. He was a very meticulous experimenter, so he had very thick walls, so absolutely no environmental noise or anything could get in. He could perfectly control what stimuli the animals were being exposed to, and he started looking at under what situations this joining together of stimuli occurs, this conditional reflexes, this adding on of a learned element to an originally reflexive behavior. And the terminology that was used was, he said initially, I mean, I'm going to use sort of terms to try and explain these things to you. So initially, Pavlov thought that there was a natural link between the brain centers in the dog's head, which identified food, and the brain centers that cause saliva production. He called food the unconditioned stimulus. It did not require learning. And he called the saliva production caused by food an unconditioned or unlearned response. He then said, when you repeatedly present another stimulus, initially neutral, initially it has no response associated with it. In this case, Pavlov. So Pavlov appears with the food, Pavlov food, Pavlov food. So you have CS and US, conditioned stimulus and unconditioned stimulus appearing together. And the dog produces saliva to the food after Pavlov gives him food. After multiple pairings, Pavlov suggested a new pathway, a new neural link rose between the conditioned stimulus and the unconditioned stimulus. And now, when Pavlov appears, that Pavlov area of the brain is activated, it passes activation through the new pathways to the food center, and the food center has built-in pathways to saliva production. So now, presence of Pavlov starts causing saliva production. As I said, this was called classical conditioning or Pavlovian conditioning, sometimes called respondent conditioning. So, first thing, Pavlov was a very active researcher, and these days we would say he destroyed several possible PhDs because he did a lot of the work himself. Um, so, what the first thing that Pavlov zoomed in on was how general is this mechanism? What multiple parts of the brain can be conditioned stimuli centers and can get linked up to the food unconditioned stimulus and then start producing a saliva response. So he tried switching on a bell, giving the animal food, bell, food, bell, food, bell, food, conditioned stimulus, unconditioned stimulus, conditioned, unconditioned, bell, food, bell, food. Pretty soon he switches on a bell, the animal starts producing saliva. He tries switching on a light. Now this is visual input, different system. Light food, light food, light food, light food. He switches on a light, saliva production, indicating it's this part, this region can also get connected. He tries touch, strokes the dog, give it food, stroke food, stroke food, stroke food. Pretty soon, stroke, saliva production. So he has identified this system as being very widespread. Um, a lot of kinds of conditioned stimuli can be linked to the unconditioned stimulus of food and start causing a conditioned response. So when the saliva is being caused by a conditioned stimulus rather than unconditioned stimulus, it is called a conditioned response, a CR. So he then tries to look at, well, what other kinds of conditioned responses are there? What kind of, apart from salivation, what other things can be conditioned? And again, he finds a broad set of involuntary responses can be conditioned. Stomach secretions can be conditioned. Heartbeat can be conditioned. He, he checks a lot of them. He then looks at what are the parameters under which the system operates. So, for example, how long a delay can you put between the conditioned and unconditioned stimulus and still cause a learning to take place, a growth of a connection between these two centers to take place. And he finds that in most cases, the time delay has to be relatively short. So, very typically, 1 or 2 seconds conditioned stimulus appears. Within 1 or 2 seconds, an unconditioned stimulus appears. And then, after repeated presentations, a learning will show itself. Longer delays, and you get poor or no conditioning. So, this seemed to be evidence for a very universal kind of associative mechanism in the brain. Any part of the brain center could seem to get linked with any kind of a reflexive response center. You could condition a lot of them. And if you think about it, it makes sense uh, in, in a particular way. If you go to a tree and you find food there, and you this happens 2 or 3 times, then it makes sense the next time you see a tree like that or that tree that you should start salivating. Okay, food is on its way, as it were. So it's a way of the system, your psychological system, picking up correspondences or co-occurrences in the world and internalizing them. But Pavlov also thought, well, you know, co-occurrences in the world happen, but sometimes they unhappen as well. So you've gone to a tree, eaten food, gone to a tree, eaten food, and it makes sense for you to look for the tree when you start salivating, you see the tree. But of course, the tree could be out of season as well. It could not be fruit season. So it makes sense for this an association built up via this mechanism to alter as well as circumstances change. So he tried to check for that, and he called this phenomenon extinction. What he would do is he would have food, food, lots of pairings. Pretty soon the association, the link in those two regions grows, and he rings a bell and the animal starts producing saliva. Now he goes into extinction trials. So the world has changed. He rings the bell, gives the animal no food. So conditioned stimulus, and then no unconditioned stimulus. Conditioned stimulus, no unconditioned stimulus. He does that several times and he finds the conditioned response, saliva, stops. So bell, no saliva. Okay. It makes sense again for this mechanism to be flexible, learning mechanism to be flexible in this way. But in terms of the underlying mechanism, Pavlov was curious what has happened. The initial learning he had suggested was due to a link appearing, a neural pathway or link appearing between two centers that were earlier not connected. What happens during extinction? There were two possibilities. One was that that link that had built up has disintegrated, it's disappeared. Pavlov, as I said, was a neurologist. He didn't think this forming of links and disintegration of links was what happening. That was the mechanism behind it. He thought it was much more likely that initially during learning, conditioned stimulus, unconditioned stimulus, food, so bell, food, a link forms. During extinction, he thought a second inhibitory link forms between the conditioned stimulus and the unconditioned stimulus. When the bell comes and there's no food, an inhibitory link formed between bell and food. And as you keep on doing extinction trials, the excitatory and inhibitory links become balanced, and that stops the saliva production. So there are two hypotheses or possibilities. Either the link has formed and then disintegrated, or the link formed and it's been counterbalanced by an inhibitory link. He had various reasons for thinking that the inhibitory links would decay faster. And so he tested this. He did learning trials, bell, bell, saliva. He did extinction trials, bell, no food, bell, no food, bell, no food, until saliva production stopped. Then he left the animal alone for multiple days. And then he brought the animal in again and rang the bell. The animal produced a few drops of saliva. Okay? And this Pavlov called spontaneous recovery. And his idea was that an inhibitory and excitatory link were balancing each other. That's why extinction had happened. He left the animal alone, no more learning trials. Both links decay, the inhibitory one faster, and the excitatory one also has decayed, but it's become slightly more strong than the inhibitory one. So now he's rang the bell, and there's a small amount of conditioned response, a small amount of saliva production that's happening. So that was his idea, understanding of extinction. He then explores the question a little bit further. He said, all right, so you can have a link forming between a conditioned and unconditioned stimulus. Can we extend this link and have a longer chain of links in the brain? And this is what he calls higher order conditioning. In higher order conditioning, he first does a standard conditioning study. Bell, food, bell, food, bell, food. Bell is the conditioned stimulus. Food is the unconditioned stimulus. Pretty soon, bell, saliva production. So that a conditioned stimulus is now producing a conditioned response. Next, he takes another stimulus, a light. So he switches on the light, rings the bell, give the animal food. Light, bell, food. Light, bell, food. So he's now got conditioned stimulus 1, the light, which comes before conditioned stimulus, bell, which comes before the unconditioned stimulus, food, and causes saliva production. After multiple trials of this second layer or second link of conditioned stimuli, he tests the light by itself. He switches on the light, the animal starts producing saliva, indicating that there is now a link between light and bell and food, and therefore there's a conditioned response. And this he calls higher order conditioning. Something like our responses to words would be explained by higher order conditioning. So you've been spanked and called stupid, stupid, spank, stupid. And pretty soon, stupid makes you feel bad. And then whenever you hear the person saying this person is stupid, you're starting to feel angry about it, because that context of stupidity, well, is now linked to the stupidity, which is linked to spanking, and that now makes you feel unpleasant about certain things. Or tone of voice, and you can say, next he's going to call me stupid, and that then is spanking. I mean, I just explained that as if you were thinking things through, but remember, conditioning mechanism is not based on thought. It's not based on subjectively available thoughts. It's a separate pathway. Certainly, dogs don't have thoughts in the way that we think about them, and they can show higher order conditioning. All right. So higher order conditioning, you have original conditioned stimulus producing a conditioned response. When you have a second conditioned stimulus linked to the first one, pretty soon the second conditioned stimulus will also start producing a response. The next question that Pavlov thought of was, is our associative system, is this linking system only becoming active in the presence of an unconditioned stimulus? यानी कि आप conditioned stimulus को unconditioned stimulus के साथ show करेंगे, only then will there be a link कि unconditioned stimulus, which is a naturally reflexive stimulus. फूड के साथ तो सलाइवा लिंक्ड है पहले से रिफ्लेक्सिवली, सिर्फ वहां पे लिंकेज होती है, या अगर आप दो कंडीशन स्टिमुलाई बार बार एक दूसरे को अपीयर करेंगे तो वो भी आपस में कनेक्ट हो जाएंगे, क्योंकि उनमें से अभी कोई किसी unconditioned stimulus के साथ लिंक नहीं. Let's say the animal is untrained and you keep showing him light bell, light bell, light bell, light bell. Animal's watching, okay, whatever. Now the question Pavlov is asking is, have these two become linked because they've been occurring together? Or does one of these conditioned stimuli need already to be linked to an unconditioned stimulus like food before the learning mechanism switches on? Now this he called sensory preconditioning. So he's just showing pairs of stimuli. These are both conditioned stimuli. इनमें से कोई unconditioned के साथ link नहीं है. Where light bell, light bell, light bell, light bell, and you don't know if there's a link has formed or not. But then he picks up one of the conditioned stimuli, bell, links it to food, bell, food, bell, food, bell, food, and pretty soon the animal is producing saliva. Now the question is, the light has never been linked with the food directly. It's only been linked with the bell. The bell has been linked with food. Did early on in their initial double presentation had a link form between them? He checks this by switching on the light. The animal shows saliva production. This showed that initially, when the light and bell, two conditioned stimuli, were being switched on together repeatedly, a link had formed between them. You saw this link when you attached one of the conditioned stimuli to an unconditioned stimulus. तो उससे अब conditioned response होना शुरू हो गया, वो pathway बन गई. पहली वाली pathway थी कि नहीं, आपने light on की, it turned out कि वो थी, वो पहले से थी. Now this again suggests to us that this associative mechanism is always working. It is always picking up which stimuli are occurring together in the environment and linking them up to one another, at least in some circumstances. So that's what sensory preconditioning seems to show. There were two other sort of significant phenomena that were identified by Pavlov. One of these was stimulus generalization. Now, what he's asking in stimulus generalization is, during associative classical conditioning aapne ki hai, a bell has been linked with food, and now the conditioned stimulus of bell produces a conditioned response of food. Has the animal learned to link that specific bell with food, or has the animal learned any kind of bell means food? Okay? Is it a generalized response or a very, very specific response? And he tests this by training an animal to link one kind of bell with food, and then he plays another kind of bell. Once the link has been learned, he plays another kind of bell to see if there's any conditioned response. And he finds conditioned responses. In fact, he finds a normal curve. So if in the center of the normal curve is the bell that the animal was actually trained on, उससे bells जितनी आप फर्क करेंगे in your testing phase, the more different the bell you use, the smaller the conditioned response. Maximum response उस bell को होता है जिस पे training हुई थी, लेकिन there's generalization. Animals will show a response to similar kind of bells. The more different you make them from the training bell, the smaller the response goes, but there is a normal curve of reaction centered around the original bell. Again, makes sense. I've found food at a particular kind of tree. If I see a similar-ish tree, it's not the same tree, but it looks pretty similar. Makes sense. There might be food there, right? Might be the same species or tree or something like that. Makes perfect sense. But the opposite also makes sense. It also makes sense for an organism to be able to learn a discrimination that, yeah, this tree and trees like it mean food, but different trees don't mean food. They don't mean food. And to check that, he looked at the ability of this system to do stimulus discrimination, to make it respond to a very narrow range of stimuli. Now, one idea that he pursued was maybe if I give an animal more and more experience with just one conditioned stimulus, the animal will learn to recognize it more and more sharply and over time stop responding to similar stimuli. कि ये हाँ अब बड़ी दफा देखा ये exactly वो वाला नहीं मुझे पता है. Again, I'm explaining it in terms of cognition, but it's a non-cognitive mechanism. The idea here is the system will start discriminating with repeated learning trials. Such as idea. He tried it, and the system kept on showing generalization, no discrimination. So animals, for thousands of times, they had seen a single bell paired with food. They kept on showing partial responses to similar signs, kind of bells. They didn't seem to discriminate. They just had a generalization curve that didn't seem to change very much with repeated attempts at learning, trials of learning. But you can do discrimination, but just simple repeated trials won't do it. He found that the animals will very rapidly learn a discrimination, but for that, he has to do extinction trials with these similar but different stimulus. So he would ring a bell, food, bell, food, bell, food, animals producing saliva. Similar bell, no food. Similar bell, no food. Original bell, food. Similar bell, no food. So for similar bells, he's having extinction trials. Once he does that, he finds animals will very rapidly learn to discriminate. So they will immediately or very rapidly stop responding to the similar bell and only start responding to the actual original bell. If you look at the actual learning curves, original ये था. Center में जिस जो original bell थी उस पर highest response आता था. उससे आप फर्क करते जाएं bells तो response कम होता जाता था. Once he's done discrimination trials, ये center bell और आपका curve यूं करके हो गया. यहाँ पे खत्म ही हो गया. पहले यूं करके था, अब यहाँ पे यूं हो गया. It's just not responding to that similar stimulus anymore. वहाँ पे zero हो गया response. So this it was thought that Pavlov has found the underlying mechanism of associative learning, which the British associationists had philosophized about, and he's explained pretty much everything that there is to explain about it. He's worked out the entire system fully. But there was one point, and exploring this point led to a very major re-understanding of what is the mechanism of classical conditioning. The original associationists had said for two ideas to get joined together in the mind, they have to occur exactly at the same time. Pavlov has found that if they occurred exactly at the same time, conditioning didn't occur very well. The conditioned stimulus had to occur a little bit before the unconditioned stimulus. So it had to anticipate the unconditioned stimulus. But his idea as to what triggered the learning was still the idea of contiguity, of closeness together in time with the two stimulus. The better the closeness, the better the learning. That was still his idea that this requires contiguity in time. Not exactly, not perfect contiguity. साथ साथ अगर तो नहीं होता था, थोड़ा सा अलग हो तो हो जाती थी conditions. That was the idea of contiguity. So Pavlov is working in early 1900s, and as I said, for 50, 60 years, people thought we figured it all out. In the 1960s, a major re-understanding takes place. And this is due to the work of two sort of psychologists, Rescorla and Wagner. Rescorla is the better known of them. Rescorla tried something else. He said, what is the engine of association formation, the engine of learning, maybe it's not contiguity, maybe it is contingency. By contingency, we mean the conditioned stimulus has to be statistically predictive of the unconditioned stimulus. If the conditioned stimulus does predict the unconditioned stimulus, then learning should occur. If it doesn't predict the other one, then learning should not occur. It's not simple pairing in time. So just because they appear together, he said, thought should maybe that's not the reason that learning happens. It will only happen when one of them predicts the other. Now, how to tease apart contiguity and contingency? What he does is um, he has he's instead of using linking with food, he has fear as the unconditioned stimulus. So he switches on a bell, gives an animal, a dog, an electric shock to the paw, and he looks at the fear response. Animals will learn this very fast. So pretty soon, as soon as the light is switched on, the animal freezes, showing fear. So it's learned the association. Now he takes a fresh group of animals, divides them into two groups, and does something else. He randomly switches on the light and the electric shock for one group. The light randomly आ रही है, electric shock randomly आ रहा जा रहा है. Now, purely by chance, sometimes the light would happen and then the electric shock. Every time that happened with the random group, he would immediately give the second group of animals a light and electric shock. So what's happening here is the number of pairings of light and electric shock in both these groups is equal. यहाँ पे randomly lights और shocks हो रहे हैं, कभी कभी light होती है फिर फौरन shock मिल जाता है. जब भी ये होता है इस ग्रुप के लिए immediately he copies that event, light on करके shock देता है. लेकिन यहाँ पे randomly बाकी सब events नहीं हो रहे, यहाँ पे random events हो रहे. He discovers that in this group, the animals learned no association between light and shock. These animals, both these groups have got equal number of pairings of light and shock. इस ग्रुप में भी वही उतनी पेयरिंग्स हैं, इस ग्रुप में भी उतनी पेयरिंग्स हैं. फर्क ये है कि यहाँ पे कंटिंजेंसी है, लाइट एक्चुअली प्रिडिक्ट्स द शॉक क्योंकि रैंडमली नहीं हो रहे. यहाँ पे कॉन्टिग्यूटी तो है, उतने ही पेयरिंग्स हुई हैं, कंटिंजेंसी नहीं है क्योंकि it's appearing by itself as well. The shock is appearing by itself as well. Now, when he does this, he immediately finds इस group में कोई learning नहीं हुई, इस group में learning हुई है. And he therefore has shown that classical conditioning happens when the probability of a shock, given that there's a light, is higher than the probability of a shock given that there's no light. So electric shocks are happening more when a light has just been switched on. They're not happening by themselves more than they're happening when a light is switched on. So the idea would be, okay, light actually increases the probability a shock is soon to follow. ये नहीं है कि the shock happens as many times without a light as it does with the light on. If that was the case, then there's no contingency, there's no statistical predictive value to a light. He finds that only when there's contingency does learning take place. In fact, animals are, he even tried this. So he would, for 10 times, he would switch on a light and give an electric shock, 10 pairings. But 20 times he would give a shock without there being a light. So in this case, 10 times the light has appeared just before the shock, but the shock has happened a lot more frequently when there has been no light. In this situation, surprisingly, or perhaps not surprisingly, animals didn't show a fear response to light. They showed a lowering of fear to light. In other words, their system has picked up when there's a light, a shock is less likely to happen than when there's no light switched on. Okay? So the classical conditioning system seems to be picking up not pairings, but statistical relationships between stimuli in the world. That's a much more sophisticated mechanism than a simple pairing mechanism. There are complications to this idea. So originally, remember, the physical mechanism that Pavlov had proposed was links grow between two brain regions. And this was very straightforward if what was happening was repeated pairing, contiguity. If there's contingency, then the mechanism has to be more sophisticated and somehow has to respond to statistical probabilities. And there is, in fact, an explanation of how simple growth of neurons can respond, neural links can respond to statistical probabilities, but that's a little bit beyond the scope of this discussion. I think Imran Rashid teaches a course that covers some of these ideas. So that's the mechanism of classical conditioning. It is the engine of classical conditioning is contingency rather than contiguity, and it's a very widespread system. It can link up lots of kinds of conditioned stimuli with unconditioned stimuli, and therefore reflexive, variety of kinds of reflexive responses. We've already seen two: saliva production is reflexive response, and fear also. Freezing due to fear is also a reflexive response. So, what kind of behavioral phenomenon can we understand with respect to this mechanism? What kind of human behaviors does it explain? Well, it explains a huge number of behaviors in terms of applications. Pretty much all of our appetites of various sorts, all of our involuntary responses, emotional responses, are all thought to be the result of natural conditioning, classical conditioning phenomena. So you're passing by MM Alam Road and you feel your stomach rumble, that would be classical conditioning. The fact that we feel hungry at certain points in the day because we have regularly had meals at those times, right? If you're a hunter-gatherer, you have your meal when you find it. But we start feeling hungry at particular points of the day. That again is a idea would be it's a classically conditioned response. Um, Sexual responses are thought to be significantly classically conditioned. In terms of applications, I think one very major application, of course, is advertisements. Advertisements try and evoke certain feelings in you and link them to a brand. So you have nice music, happy family, and you're feeling warm and fuzzy, and here's Lipton tea or some other kind of tea. Nice music, happy family, tea, blah, blah, blah. And pretty soon you start feeling your tea, family, yeah, it's nice, it's good. I don't know about you, but I've had a lot of tea in government offices where there was no happiness on my part anyway. But that's what advertising is supposed to do. It associates emotions with particular kinds of products by evoking certain feelings and repeatedly pairing them with the product. So you've got um, your conditioned stimulus, which is the brand, repeatedly being linked to pleasant feelings, and pretty soon the brand becomes a conditioned stimulus, and you feel happy about that brand. There's nothing particularly youthful about yellow colored drink. If you ask me, very nasty yellow colored drink and with a double caffeine load in it and youth. But oh, we are doing skateboarding, we're jumping off mountains, we're doing this, Mountain Dew, Mountain Dew, pretty soon Mountain Dew is like a, yeah, I feel young and energetic having it. That's the kind of thing that is explained by classical conditioning, and it's used in advertisements. That's the mechanism that they're trying to rely on. So that's a positive sensation. Of course, it's well known that fear is also a conditionable reflexive state. Um, any stimulus that happens to be around before any fear situation takes place, and you will pretty soon learn that fear. Anybody who's been in a car accident, right, so nasty car accident, next time you get into the car, you're feeling a surge of fear, or you drive through that place again, that particular intersection, and you suddenly feel a surge of fear. That would be classical conditioning. Um, Fear typically becomes more rapidly classical conditioning. एक pairing में भी आपका fear हो जाता है. Food typically takes multiple pairings. Um, the rate of learning of classical conditioning depends on the unconditional stimulus that you're dealing with. For food, it's slow. For fear, it's relatively rapid. Again, it makes biological sense. Um, there's some, so now we've talked repeatedly about biological sense, and this aspect of selectively tuned classical conditioning was explored or discovered by scientists, Garcia and Coling. Again, Garcia is better known, he's known as the Garcia effect. They were working for the US Navy in the middle of the Cold War, and they were experimenting with animals to see how they respond to acute radiation poisoning. It was thought there might be a nuclear war or something like that. And they had rats exposed to radiation. Now, one of the symptoms of acute radiation poisoning is nausea, extreme nausea. Um, they would expose animals to radiation, take them away. They noticed something curious. The animals who had got nausea immediately stopped eating the kind of food they had been given before. Now, they found this very curious because, of course, the animals had been exposed to the radiation in a particular room, in a particular cage. They never learned to fear that. Instead, they learned to avoid the food they'd had a couple of hours before getting nauseous. So Garcia and Coling decided to test it out. They used two kinds of unconditioned stimuli, nausea and pain. So in one situation, they induced nausea in animals, and for the other group of animals, they gave small electric shocks to the paw, pain, and they tried two. So these were both unconditioned stimuli. They tried two conditioned stimuli and tried to link both of them with each of these types of unconditioned stimulus. So they either gave the animal a particular taste, lemon tasting water, or they switched on an electric light. So it's either light and then nausea repeatedly, or lemon taste and then nausea repeatedly. The animal immediately learned to associate nausea with taste. It did not learn to associate nausea with light. In contrast, with electric shock, again they tried the same thing, light electric shock or lemon tasting water electric shock. In this situation, the animal rapidly learned to associate electric shock with the light, but not with the lemon taste. And they called this phenomenon biological preparedness, and they suggested some of the associative pathways are primed. We are biologically ready to pick up an association. The slightest event occurs and we pick up association. Other pathways are not biologically prepared and we tend to be averse to them. And this, if you think about it, explains the regular kind of conditioning as well. In anything, if you're switching on a bell and giving an animal food, well, yeah, you're switching on a bell, but there's also the light, there's the door, there's 15 other things that are happening. वो bell के साथ क्यों हो रहा है बार-बार? Animals seem to be more prepared to learn certain kinds of association. That was the finding. And this also has been linked to our sort of pathological fear responses, which we typically call phobias. When you look at phobias, very common ones include fear of heights, fear of snakes and spiders, fear of the sight of blood, injections or cuts. A lot of these, it makes sense that you have these are all sort of biologically prepared pathways, and that explains the vast majority of phobias. कि वहाँ पे the pathways was already primed, maybe something happened, maybe it didn't even need to happen. वो priming इतनी original preparedness is pathway कितनी strong थी, it starts causing a involuntary fear response, irrational fear response, which is what we call a phobia. So experimentally, to test this idea of phobias, um, American psychologist Watson and Rayner tested a little boy called Little Albert. Little Albert was a happy, smiley, pleasant child who was not originally afraid of anything. They classically conditioned him to fear a rat. So they would show him a rat. Initially, Albert is not afraid of the rat. But what they do is they show him the rat and then bang, a dish right behind him. Little baby gets startled and starts crying. They do it a couple of times, and sure enough, the baby starts crying at the sight of the rat. And then it generalizes, he starts crying to other fluffy kind of things, furry things. It just shows a generalized fear and phobia. Unfortunately, um, Watson and Rayner, this was in 1920s, they Little Albert didn't come back to the lab. They lost access to him. So they never actually got rid of their phobias. For all we know, nobody's been able to trace who little Albert was. For all we know, this poor kid went through his entire life hating anything furry around and being afraid of it. But in other modern versions of this experiment, it has proved possible once a fear has been classically conditioned to uncondition it. Now, how do we uncondition a phobia or any kind of a classical conditioning? We already know one technique, which is extinction. Um, with phobias, extinction doesn't work particularly well because fear is by itself a unpleasant stimulus. तो आपने बेल की और let's say electric shock दिया या कुछ did something in nasty and the animal learns to fear a bell. अब आपने बाद में इलेक्ट्रिक शॉक नहीं भी दिया या इंजेक्शन नहीं भी दिया, वो रो रो के भावा हो गया है बेल को देख के, दैट बाय इटसेल्फ इज एन अनप्लेजेंट स्टिमुलस, तो उसका एक्सटिंक्शन ट्रायल एज सच नहीं हुआ. The way to lower fear is actually a slightly different technique called counter conditioning. In counter conditioning, what you do is you have earlier on you've learned in association. Let me give you the example of counter conditioning. Counter conditioning, you're a baby, you go to a pediatrician. Pediatrician gives you an injection because it's your vaccination, and you cry and you come home. And the second time you go and he gives you an injection, you cry and come home. Third time you go, आप दरवाजे पे रोना शुरू हो गए क्योंकि क्लासिकल कंडीशनिंग हो गई with the sight and the sound. Now, extinction trial नहीं हो सकता इसका as such. Vaccination भी और करवानी ही है और आप रो रो के भागे हो गए, that by itself is an unpleasant event. What you can do is, so conditioned stimulus has become linked to the unconditioned stimulus of pain and fear. Okay. In counter conditioning, instead of waiting for an extinction, कि आप conditioned stimulus दें और unconditioned stimulus ना दें, instead of doing that, you link the conditioned stimulus to another unconditioned stimulus which has a contradictory conditioned response. So you go to a doctor and you get a lollipop. Okay? Remember, if you've been to a pediatrician always gave you a lollipop or a sweet afterwards. Why was he doing that? They've been trained to do counter conditioning. So now the doctor is linked both with injection and with lollipop. For injection, the conditioned response is crying and fear. For the lollipop, the conditioned response is smiling and happiness. And by growing this second link, you try and overcome the initial behavior. That's why it's called counter conditioning. ये conditioning हुई है, ये दूसरी conditioning हुई है. दूसरी conditioning से आप पहली वाली को counter कर रहे हैं, आप पहली वाली को extinction में directly ले जाने की कोशिश नहीं कर रहे, because that's difficult to do. And it's quite funny to actually see children. They take the lollipop and they're crying a little bit, but they're also smiling. So in a sense, both conditioned responses have happened. That's sort of the counter conditioning technique. It's similar to something that is done in adults, and which is adult treatment for phobias. And this treatment of phobias is called systematic desensitization. इसमें आप करते ये हैं कि suppose someone has a phobia, they have a fear of heights, so they are at a height and they feel the heart pounding and fear and so on, or they're even imagining going up in a lift or going up to a height and they feel the fear. So what you do in systematic desensitization is you ask people to imagine a stimulus. So ask yourself, imagine yourself being halfway up a flight of stairs. बहुत ऊंचा नहीं गए, आपने थोड़ा सा fear evoke किया, halfway up a flight of stairs. Person starts getting fearful and you say, okay, now do relaxation exercise. You teach them relaxation exercises one by one. अपने body के major muscle groups को tense up करें, hold them for a few seconds, then relax them completely and feel them relaxing. So you do that. These are relaxation exercises. So you first imagine the stimulus, tension होती है, आपको fear होता है, you make relaxation exercise करें. You do it multiple times, and now your fear stimulus is linked to both fear, which is your phobia, and relaxation. Now you up the ante. That's why it's called systematic desensitization. Now you say, don't imagine yourself halfway up a flight of stairs, imagine yourself all the way up. And again, link that with desensitization as well. So you keep on increasing the imagined fear stimulus, and at each stage, you keep on repeatedly pairing it with relaxation exercises. And that's the systematic desensitization technique, which is a major sort of application or treatment of phobia. Um, Notice in all of classical conditioning, the associative links that are being formed are between two kinds of stimuli. Okay, bell is a stimulus, food is a stimulus, and then once it gets linked, bell produces the involuntary reflexive response. So that is, in a sense, the definition, in a sense, of classical conditioning is the association of two stimuli. One of them originally neutral, one of them in with a natural link to a involuntary response. That's your definition of classical conditioning. Now we move on to other application and phenomenon to do with classical conditioning. It's not just found in humans, it's a very widespread mechanism, and it's found in all other kinds of other organisms as well. And people trained, for example, slugs. Slugs cause, they look like snails without a shell. They're sort of little black snaily things that wander around. They cause a lot of damage, agricultural damage. So psychologists tested to see if they could condition slugs. So slugs like eating carrots. As I said, they're an agricultural pest, but they repeatedly paired a bitter taste with carrots. Okay. मैंने स्लाइड में गलत लिखा हुआ है, carrots are the unconditioned stimulus, bitter taste is the conditioned stimulus. यहाँ पे गलत. So they repeatedly paired bitter taste with sorry, I've got it right. Bitter taste is the unconditioned stimulus. They learn to avoid it. Carrots are the conditioned stimulus. They repeatedly paired carrot with bitter states, and pretty soon the slugs started avoiding carrots. They learned it. Next, they did higher order conditioning. They then next linked carrots with potatoes for slugs that had already learned an association between carrots and bitter taste and nausea. And sure enough, fairly rapidly, these slugs started avoiding potatoes as well as carrots. And this has led to a number of sort of humane techniques to try and get rid of pests of various kinds, not just agricultural pests, but also in animal husbandry. So, in the US, what a lot of farmers have been doing is they inject a nausea-inducing chemical into a sheep's carcass, and they leave it near their pastures for jackals and bulls, and of course they eat it and get very sick. And the idea here is then they learn to avoid the livestock and will not carry it away or injure it. So that's the kind of application that's been done. In humans also, there is a very wide application. I've already talked about advertising, which is obviously an application, but there's medical applications as well. So one of the major treatments for cancer is chemotherapy. You inject chemicals that kill off the cancer, um, and that's the treatment. Unfortunately, the chemicals used in chemotherapy are poisons. They're toxins. And the body's response to poison is extreme nausea. Um, they're poisons. It's just that they kill off the cancerous part of the body faster than the rest of the body, which is why you use them. They kill fast growing cells, which is why you lose hair and things like that in chemotherapy. So आपने chemotherapy दी, cancer तो आपको solve हो गया, लेकिन you start feeling nauseous. Typically, cancer patients lose a lot of body weight. They become weak. They can't tolerate high doses, which they may need because that's what their cancer demands, but they just can't tolerate it because the sight of a hospital, the sight of food is making them throw up because classical conditioning is happening. So the idea here is you went to a doctor, you felt sick afterwards. The sight of the doctor is going to make you sick. The sight of the needle makes you sick. Very often you had some food earlier in the day, then you went for your chemotherapy. You had chemotherapy, now you feel nauseous at that food or even the thought of food. What you do is you use ये body की natural classical conditioning होती है, ठीक है? Conditioned stimulus था the food you ate in the morning. Unconditioned stimulus थी nausea. Pretty soon you had a pairing. You feel sick at the sight of that food. what you do in to control this is you set up an alternate conditioning agent. You give another classical, another conditioned stimulus for the associative system to attach itself again. So very commonly what they do is they'll give a glass of lemon juice to the patient and then give them chemotherapy. And the person will of course feel nauseous very much afterwards. And they'll start avoiding this. Lemon smell will start making them feel sick. They'll avoid lemon tasting food, all of it. But their other appetites are relatively preserved. उनके साथ क्लासिकल कंडीशनिंग नहीं हुई बिकॉज इस कंडीशन स्टिमुलस एक बड़ा प्रोमिनेंट सा दिया हुआ था सिस्टम को, उसके साथ एसोसिएशन फॉर्म हो गई इन द ब्रेन रीजन्स, बाकी सब बच गए. एंड पीपल हैव बीन नोन टू फॉर एग्जांपल weight loss during chemotherapy has been cut by 60-70% by using these classical conditioning techniques in some cases. Um, Final application, medical application of classical conditioning is that it's thought that the placebo effect works in part by classical conditioning. So you go to a doctor, you take a painkiller, you feel better, you go to a doctor, take pain. Pretty soon you've got pills and doctors associated with feeling better. Now you're given a placebo pill, it triggers some of the same responses in the body. Very often the same brain pathways, pain-killing pathways, pain-suppressing pathways in the brain are activated, and the placebo has a powerful effect, positive effect on pain-killing or other kinds of symptoms. So that's thought to be at least a part of the mechanism by which placebo effects produce their impact. There's also thought to be a cognitive component to how placebos produce an effect. We don't just have a brain linkage, we also have thoughts and ideas के डॉक्टर के पास जाने से बेहतरी हो जाती है. These expectations and ideas and memories are also thought to contribute to the impact of the placebo effect. So cognitive mechanism भी impact कर रहे हैं और classical conditioning mechanism is also thought to be one of the mechanisms behind that. आपकी reading में नहीं है, there's also evidence that classical conditioning can produce all other sorts of cognitive effects. So Gottfried and Dolan linked abstract art images with either peanut butter or vanilla until association formed. And then they would make people see abstract images, how fast can you recognize them, and people were very rapid at recognizing them. If they then overfed people on vanilla, जो उसका unconditioned stimulus है, उसको overfeed कर दिया आपको, when they do that and make people do the study again, their ability to see abstract images more rapidly has disappeared. It was at least partially a condition. So even your perceptual processes were partially conditionable. क्योंकि आपने जब कंडीशन किया था तो स्पीड अप हो गया था, आपने अनकंडीशन स्टिमुलस वनीला जो था उसको डिवैल्यू कर दिया by overfeeding a person, and the speeding up of perception disappears. So that's a suggestion that the associative mechanisms are fairly widespread. They don't just affect externally visible condition responses like sort of saliva production or heartbeat, something like that. They actually cause changes in the mechanisms of the mental system as well. And उसके अंदर अंदर भी they produce an effect. Fear conditioning, जो phobias के essential है, उसके कुछ, there are a number of, unlike the earlier studies of Pavlov and Garcia, which were purely behaviorally based, now we have brain imaging, and via brain imaging, biological interventions that we can do, and people have learned a lot more about these pathways. So for example, it's known that the fear pathways, which are linked to fear conditioning, pass through the amygdala. And they seem to be involving the neurons in the amygdala which respond to the neurotransmitter glutamate. With this knowledge, people try to look at whether unpleasant fear responses, phobic responses could be altered. In animals, if you give people, if you give animals a glutamate neurotransmitter blocker that stops extinction, the conditioning will keep on happening. If you give it a glutamate agonist, fear conditioning, conditioned fear, it goes into extinction more rapid. They tried the same thing with human beings. Uh, People who had a fear of heights, a phobia. अब आपके fear of heights का क्या मतलब? Fear by phobias of heights, as we'll go on to see later in the course, it's really debilitating. It means आप एक जॉब नहीं ले सकते क्योंकि उस जॉब में आपको 2 मंजिलें ऊपर काम करना पड़ेगा. आप क्लासें अपनी सिलेक्ट नहीं कर सकते एक बिल्डिंग में क्योंकि उसकी हाइट बहुत ज्यादा है. That's a kind of interference in life that phobias cause. So they looked at fear of heights in humans, and they did a double-blind study. One group of people got a drug that enhances glutamate. The other people got a placebo. Both sets of people also got desensitization, systematic desensitization. The ones who had got a glutamate agonist showed more improvement, and the improvement lasted a longer phase of their life, and their learned lowering of fear extended more and better to real world circumstances than those who had had the uh, placebo. So it shows that the glutamate pathways in the amygdala are very centrally involved in the fear system. In this case, in this example, the extinction of the fear system, a learned fear, classically conditioned fear system. Based on this, people have also attempted to look if there's some genetic involvement. And this again, we'll look at a little bit more later. There's initial findings that there is a gene that is linked to a greater fear or startle response. Startle response, of course, that's a startle response. There's a gene linked to a greater startle response, and it's linked to the reactivity of the amygdala. How, for what, at what point the amygdala switch on and show a full-blown fear response. And there are other pathways linked to this gene, including impaired cognitive control and impaired extinction of fear. But these studies are still initial, and later studies have found that वो एक gene नहीं है, there are multiple network of genes that all seem to be involved. We've looked at this idea before when we looked at genetics. For a difference in ability like this, it's far more likely to find a network of genes rather than a single gene. And that's what the initial genetic studies have shown. Right, let's stop here for now, and we will continue with learning and operant conditioning in the next lecture.